Okay. I think we can start. So I'm going to present you. Okay.
Thank you, Mark.
So it's my pleasure today to welcome you to the monthly EUROTOX webinar. So today we have the pleasure to have Carola Voss. And Carola is right now at the Hannover Medical School, and she's going to give us a presentation on NAM to study epithelial perturbation in carbon nanomaterial-induced lung inflammation. Dr. Carola Voss studied biology at the University of Leipzig, after which she undertook her postgraduate studies with Professor Roten Rustinhauser, I'm sorry for the pronunciation, in Freiburg, in Switzerland. There, she investigated the effect of cellulose nanocrystal aerosol on a triple co-culture model on the human epithelial air-blood barrier. She was awarded a postdoc mobility fellowship on the Swiss National Foundation to conduct research in stem cell engineering with Professor Wolfgang Tang and the Australian Institute for Bioengineering and Nanotechnology. So in Australia, I guess. No, in Australia. Our goal was to study a rare form of childhood hypomyelination by using direct differentiation of patient iPSC into oligodendrocyte. Quite original. Then she returned to Germany in 2017, where she worked as a senior scientist in the group of Tobias Stoger at the Helmholtz Center in Munich. So her scientific interest drove her to engineer the alveolar barrier in vitro for other primary cell lines or iPSC-derived lung progeny. So this cells model will help to decipher inflammatory processes for environmental triggers, to study also gene deficiencies on alveolar cell functionality and model cellular crosstalk into the alveolus in co-culture in 2D and 3D system. So for our efforts to develop new approach methodologies to investigate mode of action or environmental particles for respiratory health, she was accepted into the EMI Noether program of the DFG in Germany. Okay. Thank you very much, Carola. So the floor is yours. And so the last 15 minutes, I will have questions in the chat, and I will read you the question through the chat.
Thank you so much, Mark, for this really nice introduction. I'm really glad for the opportunity to share the concept that we're working on in my lab. And as you already mentioned, we are focusing on trying a new approach methodology to understand the epithelial impact of carbon nanomaterials, and of course in reference to respiratory health. And in the next couple of slides, I would like to touch upon why the study of carbon nanomaterial impact is important. Why do we need to know or how we try to know the mechanisms of actions in the lung at all, and how we can improve this understanding. So, of course, everybody knows, and it's widely mentioned, that air pollution is one of the greatest health risks in our era globally. About 6.7 million deaths, premature deaths every year occur due to the exposure of air pollution. And we can see that about 4.2 million deaths are a result of the exposure of air pollution to ambient air pollution, so the outside air. But more and more also the impact of indoor air pollution, for example, from cook stoves and combustion of fuels, for example, asks for more premature deaths globally, and this accounts for almost 3.8 million every year. Back in 2019, when I took these numbers from the WHO website, about 91% of the global population was exposed to air quality that exceeded the proposed guidelines by the WHO. And very unlucky for us, this number even rose to 99% in 2024. So basically, all of us, the whole world, is exposed to bad air quality, and we all suffer from the consequences of it. This health crisis due to the air pollution, which is also supported by the occurring climate change, is affecting literally our whole body. So there are a plethora of studies that show that There are even brain damages and also diseases supported by the exposure to air pollution. But of course, what's mostly known is the cardiovascular system that is detrimentally impacted by the air pollution. But of course, there's also emerging data supporting metabolic diseases like diabetes that can be connected to the exposure to air pollution. Nevertheless, talking about air pollution, of course, the lung and the respiratory tract in completion is the portal of entry of this air pollution into our body and therefore also suffers greatly from its exposure. As you probably know, we cannot decide what we inhale in contrast to what you ingest, you put on your skin, or the lifestyle you're leading. So we're taking about 12 to 16 breaths every minute, and that sums up to about 10 cubic meters of air that we inhale every day. And with it, of course, everything that is dispersed and carried by the air. So therefore, the lungs offer the most important interface to the environment. And this environment now carries the environmental and air pollution particles and also gases, for example, like ozone, a well-known trigger or stressor to the human lung epithelium, which is now also rising due to climate change. And when you look at this little graphic here on the side, I show you that about 3/4 of deaths that are connected to chronic lung diseases, including lung cancer and COPD, are connected to air pollution. And the same is true for ozone-connected lung diseases that lead to premature death, where the number lies around 46%, and it is rising. So, of course, this is nothing new to our lungs. Historically or even evolutionary, our lungs have been exposed to natural particle matters, including from volcano eruptions or wildfires, or even bacteria and viruses, of course. But since very, very recently, so the last couple of hundreds of years, the quality of the particles have changed rapidly. And I'm just showing a few examples how they have changed. So, for example, of course, combustion-derived particles, either from like exhaust or cars, are very important contributors to air pollution, but also intentionally inhaled air pollution like tobacco smoke, or indoor air pollution because you don't have any other means, for example, from cooking stoves, is now threatening our respiratory health. And of course, there are emerging threats like microplastic particles that have been now detected in literally any compartment in the world, as well as climate change related air quality changes, including dust storms, for example, containing a high containment of sand, for example, is a major threat and part of our very new life, basically. But of course, these are all environmentally and kind of lifestyle impacted air pollution problems. But then there is even a more new kind of quality added to this, where we have occupational exposures, for example, in the material using industry, and where you like drill or sand or cut different materials, and these dusts are released into the air, and the workers are, of course, exposed to this. But also very, very prominently, nanomaterials, who gather a lot of interest in industry for their unique properties, are produced in many different ways every day, and are, of course, not only produced and therefore released into the air, but also during the material's life cycle, of course. So another topic that I just wanted to mention to go further into this topic is the size distribution of ambient particulate matter as an example. So this can range from like 0.01, so 10 nanometers, up to 2.5 micrometers. And I wanted to show you the comparison between the mass burden and where we see that most of the contributed mass in our air pollution particularly PM2.5, is around a diameter of 0.1 to 0.5 micrometers. So most of the particles, talking about mass, are in this range. However, if you are looking into the numbers, the abundance of particles that is carried in our air, this dimension changes dramatically to like 10 to 100 nanometers. So most of the particles in terms of numbers that are carried in our air are of this very small nanometer range. So why is this so important for the respiratory health, and why am I mentioning it? It's because the way our lungs are structured is like a dichotomous tree where we have conducting airways, and they're getting smaller and smaller by diameter. The more distal you go into the alveolar compartment, excuse me, and studies have shown that due to the different physical forces that occur during this kind of tree-like structure, the particles are deposited according to their aerodynamic diameter. And actually, the deposition of the nanometer range particles is highly efficient in our alveolar space that you can see here in the red kind of graph here. So particularly where our lungs are most susceptible to the environment where the gas exchange takes place, they efficiently deposit. This is also the place where we have the slowest and the least effective removal mechanisms. Therefore, depending on the solubility of the particles, they can also stay there for quite a while. So for the respiratory epithelia, we can postulate that fine and ultrafine particles definitely represent the highest burden. To give you kind of a cell a size comparison of how actually small these particles are, when we're starting from a diameter of a hair, which is around 50 micron in diameter, going down to chemical structures like hemoglobin around 10 nanometers. This is actually where the nanomaterials that are used these days are located. So here you can see some gold nanoparticles that a colleague of mine in the past has produced in different shapes and aspect ratios and sizes, and they all are between 1 and 100 nanometer per definition, a nanomaterial. And so these also readily reach and are deposited into the alveolar space. So now we are left with the problematic that all the useful toxicity benchmarks like IC50s values and such are developed on the background of chemical toxicology. However, the situation with sterile and particularly with low toxin, low solubility particles, they behave very differently. And so these measurements often do not reflect the actual toxicity. So exposure limits or any regulatory measures are often used, often based on in vivo experiments, also in light of the problematic that we are lacking very robust in vitro situations or experimentations. So another aspect that I would like to mention in this regard is the initiation of the inflammatory response by inhaled particles. So here you can see a schematic of an alveolus, very, very easy, simple schematic, where particles are inhaled into, and then of course are also uptaken by the macrophages. They deposit at different stages at different places, and then usually neutrophils are recruited to the airspace upon this inhalation. And this has been studied really, really widely and also has been postulated in different paradigms that nanomaterials can elicit a pro-inflammatory response. And for example, I just want to mention two kind of paradigms that are most important, in my opinion, is the fiber toxicity paradigm, where the inhalation of fibrous-shaped nanomaterials leads to the phagocytosis of such, and of course, depending on their length, to a so-called frustrated phagocytosis, so where the macrophages are unable to uptake the whole fiber as it is, and they're kind of needle-like sticking out, which leads, of course, to the damage of some of the cell compartments of the macrophages, like the lysosomes, which further supports the already existing production of reactive oxygen species. And these species lead to oxidative stress and also inflammasome activation, which then in turn leads to more detrimental effects like cell apoptosis and further inflammatory cytokine releases. But this oxidative effect by reactive oxygen species production is also induced by the other paradigm I would like to mention, by surface toxicity. So a lot of particles, just due to their small size, have a high surface reactivity and produce oxidative stress in the cell. And all of this leads to the induction and sustainment of pro-inflammatory responses in the lung. So, of course, in the past, a lot of people are trying to understand this pro-inflammatory response and then the actual consequences in the lung due to the inhalation of different particles. And in recent years, the development of the AOP concept, so the adverse outcome pathway concept, has been conducted. And this is quite a useful tool in toxicology and human risk assessment with a high potential, where the AOPs are designed to provide a mechanistic representation of observed toxicological effects. So for example, the AOPs, they are usually numbered like here. They start with the so-called molecular initiating event. And in terms of nanoparticle toxicology, this is often the interaction of the particle with the biological entity, so the cell membrane or cell itself. So particle-cell interaction is an often mentioned molecular initiating event, and then the connection of different key events leads to a measurable outcome, the adverse outcome. For example, here I picked lung fibrosis. And interestingly, a lot of these AOPs also represent lung inflammation as a key event, and the connection between molecular event and the outcome by inflammation is actually widely distributed towards different AOPs. So we know that ambient particles impact respiratory health and that it leads to a lot of premature deaths globally. But actually, the very close mode of action, how it comes to this inflammatory response and how we could test this in vitro, is currently still limited, let's say. So the challenge remains that a lot of these nanomaterials are actually tested in animal models by, for example, intratracheal instillation. And one of the main measures for inflammation or lung inflammatory responses is the measurement of neutrophil influx. But of course, this neutrophil influx is caused by different mechanisms, and it is kind of a fruit salad of everything that is happening in the lung. And what would be helpful to understand the mode of actions particularly is the cell-specific response, so picking apart each fruit representative for a cell type and understanding how it contributes to this overall neutrophil influx representing lung inflammation. So this is the way that we would like to understand the mode of actions and then come up with better assays in the future. Excuse me. So we work mostly with these three different model carbon particles in our lab. For example, carbon nanoparticles. They are used as reinforcing fillers, but also often as color pigments in car tires, for example, but also in our printer ink, and are often called printex particles, actually. So we are all exposed to these kind of particles, and they can also be used as a surrogate for combustion-derived soot-like particles. On the other hand, we use carbon nanotubes due to their importance in an industrial setting and their often usage in new material and combined material, composite materials, due to their high tensile strength and also conductivity, for example, and they are cylindrical stacked nanostructures of graphene, and we use double-walled carbon nanotubes, so two of those stacked within each other, or multiple ones. And you can already see that they have different physicochemical characteristics. But just as a reminder, so these are now model particles sterilely made in the lab. However, if you look at the air particle sample from, I think this was Paris, where people collected air samples close to a big road, you can find not only agglomerates of carbon nanoparticles of spherical shape, but also carbon nanotubes of different sizes, diameters, and shapes within this combustion-derived air sample. So we are not that far off with our kind of model particles from air pollution particles. So what we did is, of course, we started instilling the different mice with these particles and compared it to a known inflammatory benchmark control like LPS. And after 12 hours or 6 days and 28 days, we took out the lungs and undertook single-cell RNA sequencing along with like classical histological analysis and such. And just to kind of introduce this technique, if you're not familiar with it already, basically what we did is out of the lung tissue, you make a single cell suspension so that you have really one cell by one cell, which are then combined one cell with a so-called bar-coded bead. This bead is carrying a unique identifier for each cell, so that you can then later distinguish which cell the transcripts came from, but also each transcript is uniquely identifiable through these barcodes that are attached to the bead. And so in the end, you have uniquely labeled transcripts of each cell that then are prepared as a library, which is basically a conventional PCR reaction. And this library can then be sequenced and all this data represented in your data analysis. This is how it then looks like when you analyze the data in a so-called UMAP, where basically this high-dimensional data is reduced to a 2D representation. Each dot represents a transcript of a cell, and you can see by the color code that I put here of the different treatments that already just by looking at the UMAP of our exposed mice that we have distinctive cell populations that are reacting to different stimuli in a different way. This is representative of about 100,000 sequenced cells and 41 different cell states and cell types. And with this data set, we tried to understand a cell-specific response pattern. Of course, we also measured the pulmonary inflammation of our mice in a very traditional way by a bronchoalveolar lavage and counting the neutrophils. And we aimed for and we also achieved a similar acute level of neutrophil infiltration at 12 hours. And that was important to us because we wanted to compare the mechanics of each inflammation, but not comparing apples with pears when the inflammatory levels are so different in an acute scenario. So we have a similar acute inflammatory potential basically in the 12-hour time point. Of course, this is much lower than the LPS-induced lung inflammation. And also you see that the materials, of course, due to their characteristics, have a different long-term effect. The carbon particles are subsiding quite quickly to base levels, whereas the carbon nanotubes are persistently recruiting neutrophils to the airspace, particularly the multi-walled carbon nanotubes that can still trigger inflammatory response after 28 days. Of course, one of the concepts of particle-cell interactions is the uptake of particles by alveolar macrophages. So this is a study from Tobias's lab a couple of years ago showing mice that have been exposed to carbon nanoparticles, and these are then cytospins from the alveolar lavage, so mostly representing alveolar macrophages. And I hope you can appreciate that during the time, I'm going to do it like this. My pointer is not working anymore. I'm sorry. according to the time, the alveolar macrophages seem blacker and blacker by the time, and around 12 to 18 hours, most of the particles are removed from the airspace, so we don't find them freely floating in the lavage anymore, but they're all contained in the macrophages. Coinciding with this, okay, it has crashed.
It's not moving anymore?
No, it's not moving anymore. Nothing is moving anymore.
You want to try to reconnect?
Actually, I can't do anything at the moment. My mouse is not working and I can't escape.
And from your mouse of the computer or the mouse outside the computer? The mouse on the computer.
The mouse on the computer.
Oh.
So I don't know what happened. Nothing moves anymore. Okay, that is bad.
Maybe you should shut down your computer. The pointer is working now. It's working now, no? We can see your arrow and your pointer.
Okay. But it's not reacting. Okay, just give me a minute. Maybe it just needs to catch its breath. I don't know.
It's really, let some time for the participant to.
I'm so sorry about this. I don't know what happened.
I mean, it's not your fault, Carola. I mean, if you see if it's not working in 20 seconds, you just shut down your computer and restart it, and we will wait. No problem.
Okay.
And I'm sure the people there, they will wait for you. And we have some questions already, so that's good. There's some hope.
There is some hope. Let's find the slide. Where am I? Here.
Excellent.
Okay, let's try to keep going. Okay, so I just tried to introduce basically the concept that alveolar macrophages are really our dustbins in the alveolar space. And we showed this by exposing the mice with carbon nanoparticles, and by the time points, by increasing time points, the macrophages seem blacker and blacker, whereas free particles are removed from the airspace. And this coincides, of course, with the recruitment of the neutrophils to the airspace. Here, for example, 12, 18 hours are marked here. The peak of neutrophil influx is around at 24 hours, where actually the particles are already kind of removed again. So, of course, we also checked in our mice with the different particles if this is true, and particularly for the carbon nanoparticles, we can appreciate those really black dots in the alveolar macrophages. But looking at the carbon nanotubes, depending on their shape and their makeup, this situation changes quite dramatically. So the double-walled carbon nanotubes, they're actually big agglomerates often, and you can find them basically distributed everywhere, but also in the alveolar macrophages. However, the multi-walled carbon nanotubes, they are uptaken in macrophages, but mostly seem to lie freely and also in single fibers throughout the alveolar spaces and also the interstitial space. This is dark field microscopy, so this is really a technique able to show single fibers of this material, which is quite nice looking at lung tissue. So, of course, we expected that also the alveolar macrophages are, in our scenario, kind of triggering the epithelial, the inflammatory response in the lung. And this is now a so-called differential abundance analysis. So basically, it is based on the UMAP that you've seen before, so this picture on the right. However, now the blue and red dots represent statistically significant differences in altered cell states. So basically, cells that have reacted the strongest upon carbon nanoparticle and tube exposure. And when we look now into the carbon nanoparticle situation, and this down here below is the alveolar macrophage cluster, you can see that transcriptionally, they do not interact very much with upon the exposure to carbon nanoparticles. So there is even a little bit of downregulation, but there is definitely no upregulation in this compartment to neither of the stressors. Compared to LPS, of course, this is a PAMP that really strongly activates macrophages via receptor interaction. And here we can see upregulation of classical inflammatory pathways. So that was our very, very first surprise, as we are all kind of convinced that the alveolar macrophage are the initiating cell of the inflammatory response. However, when we look into a different niche, so these are now alveolar epithelial cells that you can see here, is for all the particles the major hub for the reaction upon the contact to the different particles. And depending on particle type, this is of course in different levels of strength, but that was quite interesting to see. And already from this situation, we can see that nanoparticles have a very cell-specific response pattern. Okay, coming back to some old data. This is the mice that have been instilled with the CNP particles in different times. And here we measured proteins related to inflammatory responses, for example, the CXCL family 1 to 5. And now, of course, already back then, this was already known, but I wanted to show you a little example without disclosing too much of the unpublished data, that when we now look into our single cell data set, particularly zooming into the alveolar subcluster, so this is just the epithelial cells, that we can now really trace those CXCL in a cell-specific and treatment-specific manner. So I hope you can see that the CXCL 1 to 5 and such are highly expressed in the carbon nanoparticle expression, carbon nanoparticle instilled mice, but they're not expressed in the ones that have been instilled with the other two particles. And that is the power of the single cell data to really be able to deduce nanoparticle specific effects that we haven't been able to do before. So now we are setting out for kind of the new approach methodology that I was suggesting before to mirror these in vivo events in lung organoids. And I particularly want to highlight that this is mostly due to the fact that the elucidation of mode of actions in the epithelia is very tricky due to the limited robust models that we have in alveolar epithelial cell lines, for example. So they often do not represent the cell characteristics in comparison to the in vivo situation. And this is why we are trying to use lung organoids. And the idea behind this is to find signatures in the in vivo situation, mostly, of course, based on the single cell RNA sequencing data, and formulate mode of actions that then can be transferred into the lung organoid situation. And comparison of these two will kind of elucidate if these are a robust model or not. So talking about lung organoids, I want to mostly focus on the ones that are derived from primary tissue, so from adult stem cells that are taken out of living tissue. And already here you can see that we could isolate different cells from different areas of the lung, of course. And when you give them a nice cozy home in a three-dimensional matrix, due to their self-renewing capacity, they start to self-organize into three-dimensional structures or organoids. And this can happen either with or without the support of structural cells, namely fibroblasts, but often with chemical cues that we add to the media. This field is very dynamic and very constructive and very creative in always creating more and different complex lung organoids. On the left side, you can see some of the human organoids, and on the right side, some of the mouse organoids. But as I said, it's a very dynamic field, so this can be already extended with a couple of more systems. In my lab, we are mostly working with type 2 alveolar spheres that are a combination of epithelial cells supported by fibroblasts that then form these nice lung organoids. So of course, a student back in the day said, okay, now we have these perfect lung organoids and the nice single cell data showing that we have, for example, the CXCL responses in activated AT2s, as I've showed you before. And luckily, some of these markers are even upregulated in lipofibroblasts, which are the supporting cell type in the lung organoids. So of course, we tried to expose the lung organoids directly with carbon nanoparticles to measure some of these expression profiles. And this is just one representative, but very frustratingly answering qPCR results that I can show you, because basically you cannot induce any of the responses that we see in vivo by this direct interaction. So the cell circuit that we see in vivo has to be differently reflected in vitro. Another observation that we made is the emergence of so-called alveolar differentiation intermediates. They're called ADIs. And this is this little red cluster here in the alveolar epithelial compartment, which is comprised of AT2 activated, AT2s, and also type 1 cells, which are all known to make up the alveolus in the lung. And the ADIs are an interesting cell type because they gathered much attention, and us and others have shown that they are a transdifferentiating cell type between type 2 cells towards type 1 cells. And this, of course, occurs during homeostatic homeostasis in the lung, of course, when needed. However, they increasingly emerge upon lung injury. And when the lung injury is sustained over a couple of times, then these accumulate and also support the progression of the fibrotic remodeling in the lung. So these cells have been identified in a bleomycin model, which is an experimental model for lung fibrosis in mice. And these keratin 8 ADI cells are even more interesting when we look in our lung organoids, because they are also reflected in our model. So here you can see some immunofluorescence imaging of lung organoids that have been stained with keratin 8 in green here and pro-SPC in red for a marker for type 2 cells. And you can already see that we have red and green cells in these lung organoids represented, and furthermore some type 1 cells. So in the right picture, the red signal is podoplanin, which is a cell marker for type 1 epithelial cells, again combined with the ADI keratin 8 marker. So the cell model seems to be perfect to investigate the homeostatic changes that occur during lung injury. And we already showed when triggering an inflammatory response or an activation of the epithelium by treating these lung organoids with inflammatory cytokines, that the keratin 8, along with other stem cell markers like pro-SPC, changes according to the stimulus. And this really shows how plastic and also responsive the cell model can be. And just to show you why we really would like to drill down on this kind of mode of action is that we have, after 6 days of multi-volt carbon nanotube treatment, we see the emergence of these long, thin, stretched keratin 8 positive alveolar intermediates. This is the place where there should be type 1 cells or type 2 cells, but please don't confuse them with the keratin 8 marker here in the bronchial epithelium, which constitutively express keratin 8. So just in the alveolar space, this is something special. So taking all of this together now, I hope that I could show you a new approach methodology by combining single-cell RNA sequencing signatures from the in vivo situation, and we're trying to model these in an ex vivo lung organoid system from the mouse. And of course, in the future, we're planning to not only use mouse lung organoids, but also human lung organoids. So these are iPSC-derived lung organoids that you can see here in the top part. And by combining the information from these lung organoids then with data that exists from, for example, the human cell atlas and human lung cell atlas, where they comprise a lot of cell signatures from lung diseases like COPD, but also IPF, so lung fibrosis, we hope to understand how particles can initiate disease-specific and also progression signatures, and therefore understand where we could intercept the initiation and prevent the air pollution-driven emergence of further lung diseases. So this is kind of my research in a nutshell and my purpose in research in a nutshell. And before we head to the questions, I would just kind of like to switch gears for the past three slides and introduce to you the Early Career Forum of the EUROTOX, where I am part of. This is a newly formed group of young scientists that is a network, and we are particularly here for the early career researchers, so everybody who has less than 10 years of experience and is, of course, passionate about toxicological concepts, not only lung, everything. And why is this ECF so important? Because we really make the space and we are the platform for young professionals in the toxicological field. We like to connect and represent you, and also bring your ideas and your fresh energy to this field of science. And how do we do this? So we have a bunch of activities planned and also already kind of published. We are undertaking talk shows where we try to connect and network experienced professionals with the young community. We try to support the ERT courses for the young toxicology group, and we also are present and offer different events like poster walkarounds or Pop Up Your Poison during the EUROTOX Congress. And if you're interested to join the ECF, we are currently 8 members in the forum, but of course the community can grow by just signing up for our mailing list, and then you will be directly informed about our newest activities. This is basically the board of the ECF, or the ECF members, Olden Foran Eliška as our chair, and otherwise we are kind of distributed amongst Europe, and we would be happy to get in contact with you. So just scan the QR code and then you can join our mailing list and participate in all the activities that we are planning. And with this, I really like to thank you now for your attention. I apologize for the little technical glitch. I thank, of course, all the people that are involved in the past, but also now my group in Hanover very, very actively. And I'm particularly enjoying this old and new lab kind of situation where we collaborate intensively about this single cell data, for example. This is the ECF back in 2024, and I just copied again the QR code for your reference. And of course, I thank the funding partners for supporting my research, and now I am happy to take questions. Thank you so much.
Thank you very much, Carola. It was a very interesting and very deep talk, I would say. I was very impressed by the single cell analysis and bring a lot of information. So I have one question from Sadia Kernen-Romer. She was interested by the AOP slide. Notice that on the AOP slide, they have the key event epithelial activation occur after the key event Th2 cell activation. And so she commented that in interleukin 33 is known to play a role in Th2 activation, is produced by damaged epithelial cells. So the question is why the Th2 is before the epithelial cells. You know?
Yeah, so very good question. So I can only comment in a kind of limited way because we are, or not we, all of the people that are contributing to these AOP concept are measuring different endpoints in different kind of exposure scenarios. And of course, the key events do not necessarily have to be sequentially correct. So it's just at the time point of the measurement after a certain exposure that IL-33, for example, was released, but that does not mean it has not been released before or it's not released after. So I think this consequence or the sequence that I showed is a consequence of the experimental setup, but it is probably to an extent also interchangeably. So that is, I think, all that I can say because the AOP concept really tries to harmonize results, not going into detail about actually the sequence of events.
Yeah, because I am from Laksha study also. We are in the field of skin sensitization, and skin sensitization are one of the first AOP that has been published with the clear kinetic of event. You see, so it were very well organized, and we have in vitro tests behind this. So that's why sometimes it's not very. And our answer is very interesting because sometimes the hierarchy is not very well understood in
Yes,
AOP. So.
absolutely. And I think this is supported by the lack of really robust in vitro assays. So we all, like most of these data stems from in vivo experiments. And then of course you cannot measure every day everything. And so it's quite complicated to unravel the sequence of events. Yeah.
And me, I've got a question about the single cell analysis and because I mean, okay, in the first picture, so it's okay, it's a nice overview, and then you go more deeply into the results, and you suggest at one point, maybe I'm wrong, yeah, you suggest that there is some sort of a signature, so the cell signatures. I mean, the CNP versus the carbon nanotubes, it seems they don't act at the same cell level.
Yes,
There are
exactly.
some cells, there are common cells, and also
Yes,
some specific cells. Can you
exactly.
a bit discuss this point because I just want to have more information. So
Yes. So the reason I haven't shown this more in detail because it's still unpublished. So
okay,
I'm trying to, but I can comment on this, of course. So we have seen that for each particle and tube that we used, we started with the same acute inflammatory response, but the quality was completely different. So for example, we also conducted BAL proteomics, where we looked which cytokines are released into the airspace, and this can also be very specifically attributed to the different particles. So there are some overlapping between the two CNTs, but they are very different to the CNPs. So it's very interesting that the same kind of endpoint, neutrophil influx, has different ways or different qualities behind it. And so then, of course, the different cytokines that we measured also on the transcriptional level can then be traced back to the cells that are responsible for this. And then, of course, again, CNPs are mostly, for example, active over activated AT2s, whereas the tubes mostly act on fibroblast and macrophage subniches. So that was quite interesting. And now we're really taking each cell type, each cytokine profile, each kind of sequence in this 12 hours, 6 days, 28 days, and try to connect what is happening depending on the material. And that's very different.
and so I have a second question behind this is that okay, so this kind of specificity, okay, do you think it is due to the surface area reaction with some type of cells, or is it due to the deposition of the tubes or the nanopower, the particles? They don't go at the same sites, you see. So
Yeah.
sometimes the double or the mono well carbon nanotube can go in this site. This is why the cells are activated, and the CNP go in another site. So is it due to the spectrum of deposition, or is it due to specific reaction with the cells?
I think both. I think it's really both. Because yes, I mean, we already see with the bright field images and also dark field images, you're absolutely right. The deposition and also the particle quality is very different. But then also, for example, talking about cell death, we saw that depending on the different particles, we see increasing amounts of cell death, for example, in the multi-walled carbon nanotubes that has not been described in that let's say clarity in the publication so far. And so we can now really deduce, okay, multi-walled carbon nanotubes have a completely different mode of action via more cell damage, whereas carbon nanoparticles are uptaken but still are able to activate somehow the epithelium, and the double-walled carbon nanotubes are actually in between, and they are the most mysterious because they kind of offer a spectrum of both. So that's like a little bit overlapping with the carbon nanoparticles and also with the multi-walled tubes.
But I guess with your organoids and your new cell assay, you will have more information on the
We
single
hope
cell
so.
level.
Yeah, we hope so.
And so I've got a long comment and question from one colleague. So great talk. I noticed that the corona effect is never mentioned, notably on the slide presenting the initiation of inflammatory response, fiber versus surface toxicity. Do you think the differences observed in vivo and in vitro may result from different corona? First question.
Yeah. Okay. I know that the discussion about the particle corona is very big and has been big in the past. We acknowledge this, but we have not gone into analysis of this part. However, to disclose fully, we disperse our particles in lung surfactant actually. So basically, we're not using any artificial or protein like FBS or so, but we're trying to use a representative liquid or fluid, and that's why we're using a lung surfactant that has been retrieved and isolated from pig lungs, basically. And this is our dispersion medium, and we hope that we kind of nivellate the corona aspect in this regard. But of course, putting it into a mouse lung versus putting it into a cell culture media will always change this corona dramatically. Yes, there will be an influence. Yes, for sure.
Yeah, we have the same issue with our silica nanoparticles, amorphous silica nanoparticles. So I continue the. So it is possible to expose organoids with different CNT previously exposed to pulmonary surfactant. That's what you said. For example, to mimic somehow the corona parameters. So that was exactly what you said. Okay. And so, but don't you think there is some sort of a dynamic exchange between the protein or the lung surfactant? And sometimes you have some particles can be free at some time, and then these particles are reacting because we are putting a lot of particles in the mind, a lot of particles in the assay. But there is a dynamic exchange between. So I'm not 100% sure that all the particles are always covered by the corona.
Yeah.
What do you have a comment on that?
Well, yes, I understand where you're coming from. However, also the protein content is usually super high in the in vitro situation. So even if they kind of dynamically exchange, I don't think it's exhausted. So they will always kind of be surrounded by proteins. But since for example, the alveolar fluid is also really high in protein content. I think this is not, of course, the composition is different, but the dynamics behind it might not be so different. Yeah, so there's also lots of protein in the natural situation. And coming back to a very technically and experimentally driven answer, we can't really change it. So for example, the lung organoids, they have a very, very, very low protein-based media. So that's actually something that we like about the system. There's other drawbacks, of course, but conventional 2D culture, some of these cells do not survive without the proteins. And then, of course, it's also not meaningful to do that. No? It's
When
just
you
a
have
limitation.
when you have 10% serum, that's a lot of protein.
Yeah.
And there's a lot of lipoproteins also, which has that's also an issue. Okay. So me, I've got I've got one more question. It's about the chemokine. So I mean, how can you explain this so specific profile of chemokine? The CNP are producing chemokine and not the others. Do you have any explanation? Because we know that Okay. Do you have any kind of explanation?
Well, I mean, we were actually struck by that it's so specific. And to unravel this, I think we should first try to have a robust in vitro model because then you can really kind of understand the interaction. But we already noticed going from the in vivo situation to the in vitro situation, we lose specificity. So we can elicit CXCL1 release, for example, but it's then released by all particles, by all particle treatments. And that was kind of striking because that doesn't happen in the carbon nanotube fraction in the mouse, for example. So the closest answer that pops into your head is, of course, well, the immune system is missing in the in vitro situation, and it's there in the in vivo situation. So the dynamic of the inflammatory response and the presence of the immune system must modulate whatever is happening very locally and then systemically over time. But I cannot answer more than that. And we already discussed like the different cell particle interactions that are also, of course, supporting this very specific response pattern.
Okay, thank you. So we have no more. Oh, okay. I've got one more question. This is a question about NAMs. Okay, to create new NAMs, is it necessary to use approaches in animal model? Question. This means that animal experimentation is necessary for NAMs?
I don't think so. So I think this is the road I chose, but there are also quite other approaches where you start by all the existing data and you try to deduce sequences or molecular initiating events via computational tools, AI and such, or you start with slices of animal experimentations that already have been done. You start imaging analysis and stuff like that. So I think there's lots of activity at the moment to be not dependent on animal experimentations. It's just the road that I chose to come up with better in vitro system, but not all NAMs are in vitro systems. Can be something else. So I think there's also other ways to go about.
Okay. I think there's no more question, and we are just arriving at 2 o'clock. That was perfect. So thank you very much, Carola. That was a very interesting presentation. I'm very impressed by the single cell analysis. It's a very nice, well-conducted technology. So it's really
Yeah.
providing a lot of information, and the way you're mapping with the different type of cells is also very, very appealing. I mean, it's very, you can see it very well.
Yes. Thank you so much.
That's great. Okay, so thank you very much, and thank you for all the participants, and see you next month for the next webinar. Thank you. Bye-bye. Have a nice day.
Thank you. Bye. Thank you. You too.